This is Lecture 12, Part 2A. This is the topic of aerobic respiration, and in this part we're going to focus on the citric acid cycle, which is also known as the Krebs cycle, and the tri-carboxylic acid cycle. In our previous lecture, we started talking about aerobic, meaning in oxygen, or with oxygen, respiration of glucose. A very important catabolic process that occurs in cells and is used to make ATP. That equation looks like this: C6H12O6. This is glucose. It's a very high energy molecule. It has many available carbons that it can donate in chemical reactions. Glucose is reacted with 6 oxygen, and that is converted to 6 CO2 plus 6 water plus energy. Okay, so this is a catabolic, spontaneous, exothermic reaction. And in this reaction, the electron donor, this is a redox reaction, is glucose, okay, up here. And the electron acceptor is oxygen, up here. The electron donor and the electron acceptor are always shown on the left-hand side by tradition in a fully balanced redox reaction. Let's consider the various stages of aerobic respiration next. Aerobic respiration takes place over 3 successive biochemical stages. The first is glycolysis. We discussed that last day, and we'll do a short recap in the next moment. The second is pyruvate processing and the Krebs or citric acid cycle. That's what we're going to focus on in this part 2A of this lecture. And then the third and final stage is the electron transport and oxidative phosphorylation. The first 2 stages, glycolysis and the Krebs cycle, are connected to the final stage by the production and movement of reduced coenzymes, specifically NADH, here, and FADH2. All of these, in the context of aerobic respiration, are going to end up contributing to something called the electron transport chain, which makes oxidative phosphorylation possible. During each of the first 2 phases, ATP is made by substrate level phosphorylation. In the final stage, ATP is made by oxidative phosphorylation. Let's recap a little bit about glycolysis now. Glycolysis, which is the breaking of sugar, hence the name, glyco for sugar, lytic for breaking, occurs over 10 biochemical reactions. It is regulated at the 3rd reaction. As I described last day, you should know the enzyme that plays a role at the 3rd reaction and what it catalyzes specifically. Many of these reactions are redox reactions, but not all, and I discussed that in the previous lecture. Glycolysis occurs in the cytoplasm of all cells, both eukaryotic and prokaryotic. During glycolysis, glucose is successively oxidized to pyruvate. 1 glucose produces 2 pyruvates. All of the carbons originally present in glucose are conserved and carry on to the next stage of aerobic respiration, which is the Krebs cycle. Some ATP is used, but more is generated, so there is a net gain of ATP. And we also have the production of reduced coenzymes, specifically NADH, which is then sent off to the electron transport chain. Let's do a little accounting of glycolysis. In other words, let's think about what molecules go in, what comes out, and where the molecules that are coming out end up. 1 6-carbon glucose goes into glycolysis. 2 3-carbon pyruvates come out. They go off to the Krebs cycle. 2 ATP go in, 2 ADP go out, 4 ADP go in, and 4 ATP come out. This means we have a net gain of 2 ATP, and those ATP are made by substrate level phosphorylation. 2 oxidized NADs go in, 2 reduced NADHs go out. These go off to the electron transport chain. Okay? The ADP would just go off to be used to make ATP at other reactions. For example, they may get recycled through glycolysis again. The ATP go off to do cell work. That's an accounting of what happens during the glycolytic reactions. The Krebs cycle, which is also called the citric acid cycle or the tricarboxylic acid cycle, occurs in the mitochondrial matrix of eukaryotes. So in eukaryotes, this is happening in the mitochondrial matrix, and in prokaryotes, this is happening in the cytoplasm, like glycolysis. The Krebs cycle process only occurs if oxygen is present. Otherwise, glycolysis will simply continue, and I'll talk about that a bit more in the next part of this lecture. And what will happen is the pyruvate will enter the mitochondria. It does that, remember, pyruvate is a polar molecule. It's a neutral but polar molecule. Therefore, to enter the mitochondria in order to get to the mitochondrial matrix, it relies on transport by a carrier protein. It can't simply passively diffuse across both the outer and the inner mitochondrial matrix. As soon as it gets into the mitochondria, into the matrix, it will participate in something called the preparatory reactions, which we'll look at next. And after that, it will continue on into to participate in the full Krebs cycle. Shown here is the preparatory reaction or reactions that pyruvate undergoes in order to enter the Krebs cycle. For every 1 glucose, you have 2 pyruvate. This pathway is shown from the perspective of a single pyruvate, but you need to keep that in mind. So the pyruvate is shown here in purple. The pyruvate is converted to a molecule called acetyl-CoA. In order to do that, it is reacted with another molecule called coenzyme A. As that happens, reduced nicotinamide adenine dinucleotide, or reduced coenzyme, is produced. That's going to go off to the electron transport chain. Another thing that happens is 1 molecule of CO2 is produced, and that accounts for 1 of the 3 carbons that are present in pyruvate. The acetyl-CoA is then reacted with a molecule called oxaloacetate, and that is a molecule that is part of the Krebs cycle. It's a Krebs cycle intermediate. That then converts this entire structure to something called citrate, and that reaction actually officially begins the Krebs cycle. All of these reactions are catalyzed by an enzyme complex called pyruvate dehydrogenase. It's a 3-protein enzyme complex. Okay? And that pyruvate dehydrogenase mediates each of these steps, both of these steps, allowing the conversion of pyruvate to acetyl-CoA and allowing it then to react with oxaloacetate to form citrate. Now let's look at the Krebs cycle. Shown here is the entire Krebs cycle. What we just considered, the preparatory reactions, are occurring here. There's pyruvate, here's the reaction with acetyl-CoA, or to form acetyl-CoA, it reacts with oxaloacetate, shown here, and it becomes citrate, and then officially enters the Krebs cycle. Remember, up here at this stage, we lost one CO2, and we generated one NADH. So now what happens from the point of view of citrate onwards is the Krebs cycle, or the citric acid cycle, turns. A cycle differs from a pathway in that it ends and begins with the same molecular structure, whereas a pathway begins and ends with two different molecular structures. In the glycolytic pathway, we begin with glucose and we end with pyruvate. In the Krebs cycle, officially we begin with citrate and we end with citrate. So there are several biochemical reactions that are occurring in the Krebs cycle. Everywhere you see an arrow, there is a specific enzyme that is catalyzing that reaction. Some of these reactions are redox reactions. So anywhere that you see oxidized NAD+ entering, so here, here, or oxidized FAD entering, shown here, and you see those oxidized coenzymes getting reduced to form the reduced structures NADH or FADH2, then you know that a redox reaction has occurred at that point. The other reactions fall into different biochemical categories. As the cycle turns, some ATP is made. That ATP production is shown down here, and that is mediated first by formation of a high-energy molecule called GTP. GTP is guanine, triphosphate, and it is once formed, then used to make ATP, adenine triphosphate. This is a formation of ATP by what is called substrate level phosphorylation, which I have described for you previously in class. I want to point out that each of these reduced coenzymes, the NADH and the FADH2, from the perspective of aerobic respiration, are going to go off to the electron transport chain. So that's going to go off to the electron transport chain. That's going off to the electron transport chain. That's going off to the electron transport chain. And that's going off to the electron transport chain, as did this. Okay? I also want to point out the carbons. Remember that each pyruvate is a 3-carbon molecule, and that for each one glucose there were 2 pyruvates formed. Each pyruvate is fully oxidized to carbon dioxide. 1 carbon dioxide was produced in the original preparatory reactions, a 2nd carbon dioxide is produced here, and a 3rd carbon dioxide is produced here. So by the time we have gotten to the succinyl-CoA step of the Krebs cycle, we have all of the carbons in pyruvate fully oxidized to produce CO2. The fate of that CO2 is to diffuse out of the mitochondria and then out of the cell and ultimately out of the organism. It is simply lost as a byproduct or a waste product of aerobic respiration. At this point, all of the electrons that were present originally on glucose are now found on reduced coenzymes, which are traveling off to the electron transport chain. So if we think about what goes in and what comes out of the Krebs cycle, 1 pyruvate went in and 3 carbon dioxide came out. Its fate is to diffuse out of the cell. Okay? One guanine diphosphate and one ADP went into the cycle. One GTP came out, which was immediately used to make one ATP by substrate level phosphorylation. One flavin adenine dinucleotide in the oxidized form, shown here, went in, and one FADH2, the reduced form, came out. That's going off to the electron transport chain. 4 oxidized molecules of NAD plus went in, and again, this includes the preparatory reactions, this accounting, and 4 reduced NADH come out. Those also go off to the electron transport chain. So we've accomplished complete oxidation of pyrite. We have gained 1 ATP via substrate level phosphorylation, and we've also obtained numerous molecules of reduced coenzymes, which can go to the electron transport chain and contribute to its function, which we will consider in the next part of this lecture. All right, now for a couple of study hints. You need to know where all events of aerobic respiration occur. Okay, that's fairly important. You need to know the complete equation for aerobic respiration, okay, including what the electron donor is and what the electron acceptor. You need to know the accounting. In other words, what goes in and what comes out for each stage. Okay? And then you need to know some supporting details. For example, what are the preparatory reactions? And where is glycolysis regulated? Okay, and you also need to know whether ATP production is via substrate level phosphorylation or oxidative phosphorylation, and you should know the definitions of those. These are some examples of ways to question yourself as you take detailed notes on the topics covered here. Let's move on now and talk about the electron transport chain and oxidative phosphorylation.